Okay, today we're going to talk about the innate immune system, a topic which I think is fascinating and vitally important to biomedical engineers and anyone studying biomedical devices because it affects biocompatibility heavily. In fact, when most people do their jobs and a device is packaged, prepared, and implanted under sterile conditions, there are no antigens present that would elicit an adaptive immune response. So the entire foreign body reaction and the response to biomaterials is due to the innate immune response. There are a number of key features of the innate immune system. The first is the maintenance of an epithelial barrier at the skin, GI tract, and respiratory linings that acts as the first line of defense for any microbes or pathogen that enter your body. And it makes sense that it's located at these locations because these are where we're going to encounter pathogens. Okay, we're going to breathe them in, we're going to touch things, they're going to come into contact with us through these layers. And it's not just a physical layer too, they actually include chemicals, particularly in the respiratory lining, that are both antimicrobial and antiviral. The second key function is for the innate response to serve as the initial reaction to microbes and pathogens. The second key function of the innate response is that it serves as an initial reaction to microbes and pathogens. As we learned previously, this might be just on the order of minutes to hours, whereas the adaptive immune response can take a few days to mount a sufficient defense. Right? So it's particularly important for really fast infections where you need a response to simply keep it in check in order to mount a sufficient defense. Third key function is that it eliminates damaged cells and initiates the process of tissue repair. Again, this is particularly important for biomaterials because very often we might have sterile products that won't elicit an adaptive immune response at all, yet the innate immune system is still going to respond. Right? You're inevitably creating tissue damage by inserting something into the body or modifying it, and by creating that damage, the body's still going to respond. Furthermore, the innate immune response works in tandem with the adaptive immune response by actually stimulating it and making the adaptive system more effective. Even the nature of the way the innate system reacts, whether it's a bacterial infection on the outside of a cell or if it's viruses that infect inside the cell, innate immune responses are going to behave differently and thus stimulate different aspects of the adaptive immune response. Lastly, the innate immune system can react in two key primary ways. One is through inflammation, which is the recruitment of blood cells to a site of an infection, and the second is through an antiviral defense, which doesn't necessarily have to come with inflammation or massive recruitment of cells, as we're going to learn about later in the chapter. Now, there are a number of key differences between the innate and adaptive immune response. First, no function of the innate immune response requires any prior exposure to a micropathogen or antigen. That means that all the effector cells of the innate immune system are fully functional before an infection occurs or are rapidly activated by microbes to prevent, control, or eliminate infections. In contrast, the adaptive immune response can take several days to mount a sufficient defense against a pathogen. The next 2 points highlight two fundamental differences between the innate and adaptive immune response. The first being memory. Okay, there's no appreciable change in the quality, magnitude, or speed of a response of the innate immune system because it never changes. It never remembers a microbe or pathogens or antigens. It always performs its functions the exact same way in a secondary or tertiary response. All right, this is not the same for adaptive immune systems that after the first infection, they can mount a much more effective secondary response or tertiary response. The other key feature is specificity. The adaptive immune response can recognize tens of millions of different antigens. In contrast, the innate immune response can only recognize about 1,000 different generic signature traits of microbes or damaged cells. Okay, so very limited diversity. Biologically, the receptors of the innate immune cells also function very differently from that of the lymphocytes of the adaptive immune response. Next, let's talk about the way the cells of the innate immune response recognize microbes. So I mentioned before that there's a limited and not very diverse array of molecular patterns that these cells recognize. But we can kind of divide them up into two different categories. The first being pathogen-associated molecular patterns. So these are patterns that are common, unique to, or just simply appear in higher concentrations in pathogens than they do in normal mammalian cells. Okay, a couple examples could be double-stranded RNA, which is unique to viruses, Bacteria might have unmethylated DNA sequences. There could be complex lipids or carbohydrates that are synthesized by microbes, but not by mammalian cells, like lipopolysaccharides, LPS, that's one we're going to see a lot. Lipoteichoic acid in gram-positive bacteria, oligosaccharides. So these are very common to pathogens and not to human cells. Now, I think it's particularly clever that we've managed to evolve to recognize these traits of pathogens because they are themselves unable to get rid of these traits through evolution. Double-stranded RNA is vital for viruses to survive. They cannot survive on single-stranded RNA. And likewise, LPS and lipoteichoic acid are necessary structural components of bacterial cell walls. So they cannot survive in a system where they can't produce it. So despite the lack of diversity, we'll always be able to rely on these techniques. The second class of molecules that the innate immune system responds to are called damage-associated molecular patterns. So we start off with PAMPs, now we have DAMPs. Now these could be the result of cell damage through infections by pathogens, or they could also indicate sterile injury to cells for trauma, burns, chemical toxins, a few number of reasons. And what's important about this is that these damage-associated molecular patterns are generally not released during apoptosis. Right? Apoptosis is a perfectly normal cell cycle occurrence, so you wouldn't want to initiate an immune response simply from that. Okay? This is usually a result of cells having some kind of injury or damage, and thus try to alert the immune system that something's going on. Now, in order to recognize these patterns, cells have pattern recognition receptors in order to recognize PAMPs and DAMPs. Cells express these pattern recognition receptors at various parts of the cell, such as the cell surface, intracellular vesicles, and even the cytosol, because this is all areas where you might have a virus or a bacteria. Now, most importantly, the cells of the innate immune system do not react against normal, healthy cells. This occurs due to three primary reasons. The first is that healthy cells do not produce ligands for the innate immune receptors. Second, for those receptors that might possibly react against host ligands, they're located in intracellular compartments where they should encounter the host molecules unless something was wrong. And third, there's also regulatory proteins expressed by healthy cells which prevent activation of various components of innate immunity. Now, I want to take a little sidebar and talk about how those receptors are coded, because it's another key difference between the innate and adaptive immune system. Now, as I previously mentioned, the receptors for the innate immune cells have a very limited diversity. And that's in part because they're encoded by inherited and intact genes. Okay, so it results in recurring non-diverse receptors that are non-clonally distributed amongst all cells. And because of this, we only have about 100 different pattern recognition receptors. Now, at one point, this was known. It was also known that adaptive immune cells, lymphocytes, can recognize tens of millions of different types of antigens. Yet, we only have around 20,000 different genes. So how is it that these cells can make such a diverse array of receptors from such a limited pool of genes? And then it was discovered that lymphocytes and other cells of the adaptive immune response generate receptors not from intact genes, from gene segments, okay, through a process called somatic recombination. So it's really just a juggling of the different Lego pieces of these genes to kind of create new gene combinations that can then create brand new clonally distributed receptors on individual lymphocytes. And because of this, the person who discovered it, a Japanese researcher at MIT named Susumu Tonegawa, received the 1987 Nobel Prize in Medicine. Now, returning to these pattern recognition receptors, the first class of the receptors we're going to talk about are toll-like receptors. Now, these receptors actually got their name after a compound that was first found in fruit flies called tolls. And then it was found that these compounds have very similar analogous forms in these receptors in humans, hence the name toll-like receptors. Now, there are 9 different varieties, and they recognize a lot of molecules associated with pathogens as opposed to mammalian cells. So many that we mentioned before, LPS, lipoteichoic acid, flagellin, which is the material that, or the mechanism that bacteria use to propel themselves, double-stranded DNA, and the like for viruses. 2 major toll-like receptors that we're going to see pop up often are toll receptor 2 and toll receptor 4. So toll receptor 2 will recognize the lipoteichoic acid toll receptor 4 will recognize LPS. In fact, they also recognize, these two in particular, recognize endogenous molecules that healthy cells make, but whose expression and location, like in the cytoplasm or in the extracellular space, would indicate damage to the cell. For instance, heat shock proteins or high mobility group OX1, which is a protein involved in DNA repair. So if these are found outside the cell, it's a big clue that that cell has been damaged, and that these proteins normally in the cytoplasm have been released into the extracellular space. So that's why they activate these cells. Now, as the picture indicates, there's a number of toll-like receptors that are expressed on the cell membrane and also in the intracellular membranes of these vesicles. And usually it has to do with how microbes or pathogens are processed. For instance, many of the compounds that are recognized in the intracellular compartments or vesicles are products of phagocytosis. So if these cells were to break down pathogens or viruses, they would find themselves in the intracellular space, hence why you need the toll-like receptors on the inside that recognize things like double-stranded RNA or DNA, products of viruses. Then, once a toll-like receptor recognizes these antigens, it activates signaling pathways and transcription factors that induce the expression of genes, which eventually help express cytokines that are important for the inflammatory and antiviral responses. Now, toll-like receptors are examples of membrane-bound receptors. Okay, they're found in the outer membrane of a cell, they're found in the intracellular membranes of endosomes, but there's also a class of receptors that exist just in the cytosol. And the ability of the innate immune system to detect infections in the cytosol is important because parts of the normal life cycle of some microbes takes place in the cytosol. In fact, some pathogens have even evolved mechanisms that enable them to escape the phagocytic vesicles to the cytosol, which means they can escape the cell's eating mechanisms. I won't go into too many details about these classes of receptors, but I want to hit on a couple of them. So first one is a class called NOD-like receptors. The two most heavily studied NOD-like receptors are NOD1 and NOD2. NOD1 binds to diamino-pimelic acid, DAP, and NOD2 binds to myramyl dipeptides, MDPs. Now, NOD2 is important because it's found in high concentrations in cells of the epithelial layer of the small bowel. And genetic mutations of this particular receptor increases the risk of the autoimmune disease Crohn's disease. The second class of receptors are called RIG-like receptors, which bind to viral RNA, and because they're linked to viruses, they're important for the production of type 1 interferons, one of the body's main ways of fighting viruses. The third class is called cytosolic DNA sensors, okay, another mechanism to recognize double-stranded DNA in the cytosol. These sensors help initiate antimicrobial responses. Now, epithelial barriers are a huge component of innate immunity. They exist in gastrointestinal and respiratory mucosal surfaces and the skins of mammals, and they act as the main interface between the environment and the host. So the defense in this case is primarily a physical barrier. So with the exception of the skin, they exist as continual monolayers, so single cell layers connected via tight junctions. Okay, tight junctions don't exist anywhere else in the body except for these epithelial layers and also the blood-brain barrier, but we're not going to get into that. These tight junctions eliminate the possibility of paracellular transport, that is, passage around the cells. Okay, so pathogens, because of the tight junctions, only have the transcellular route option going through cells, or they have to disrupt the tight junctions. Furthermore, and again with the exception of the skin, these epithelial layers have a mucus layer on top of them that acts as a diffusion inhibitor. I actually found out that the body produces around a liter of mucus a day that continuously gets reabsorbed into the body and generated again. Gross. Epithelial barriers also have chemical mechanisms for fighting against microbes. Okay, the two big classes of chemicals that help do this are defensins, which are small peptides produced by epithelial cells and granule-containing leukocytes that can disrupt functions of the microbial membranes, and then cathelicidins, which are peptides produced by epithelial cells and neutrophils that have a broad range of mechanisms, including the direct toxicity to a broad range of microorganisms. And also they serve to activate and recruit leukocytes and other type of cells for the eradication of microbes. Another component of the epithelial barriers is that they have a type of T lymphocyte called intraepithelial T lymphocytes. So these are continuously present in these barriers. They're not circulating. And while they do have a typical T-like receptor, a T cell receptor that we're going to talk about in later chapters, these cells in contrast to circulating T cells of the adaptive immune system, typically have a limited diversity of their antigen receptors. And they recognize a small number of commonly encountered microbial structures and feature innate pattern recognition receptors that we've described previously of the innate immune system. Next, we're going to talk about phagocytes, which are the first line of defense against microbes that manage to breach the epithelial barriers. They include macrophages that are always present in tissue, like tissue-resident macrophages, and then circulating monocytes and neutrophils. Now, the essential role of phagocytes is really highlighted in individuals that lack them, either through genetic disorders that prevent their differentiation and formation in the body, or perhaps also cancer patients that, through treatments of chemotherapy and radiation, which can utterly destroy white blood cells, how susceptible they are to lethal bacterial and fungal infections. So the first phagocyte I want to talk about is my favorite phagocyte, the dendritic cells. Okay, these are often called the brains of the immune system for a number of reasons, some of which we'll talk about today, and others we'll get into in later chapters. So these can be tissue-resident antigen-presenting cells that they do have minor phagocytic capabilities, which means they are capable of eating cells, but not necessarily for the direct purpose of eliminating the microbes. It's really for a way of processing the microbes so they can present those antigens. Another reason why they're so special is that they express more different types of toll-like receptors and cytoplasmic recognition receptors than any other type of cell. So it really highlights their diversity. The plasmacyto subset of dendritic cells are a major producer of the body's primary defense against viruses, which is the antiviral cytokine type 1 interferons. And because of this, because of their need to fight against viruses, they express abundant amounts of intracellular toll-like receptors, which are 3, 7, 8, 9. So I always remember the 7, 8, 9, and then the 3 is the one that stands out. So then the membrane receptors are 1, 2, 4, 5, and 6. In addition to that, they have many cytosolic RNA and DNA receptors. These are ones apt for recognizing viruses and viral compounds. Now, the ability of dendritic cells to react quickly in terms of the innate immune response, and then activate T cells later and subsequent adaptive immune responses is the major bridge between the two immune systems. Okay, so dendritic cells really facilitate that more than any other cell type. Just one of the few reasons what makes them so special. The next cell type we're going to talk about are innate lymphoid cells. Like their adaptive siblings, these cells originate from common lymphoid precursors, and they do exhibit lymphocyte morphology, yet they do not express the clonally distributed diverse antigen receptors of fully mature B and T cells. So while we say they are lymphoid cells, they are not lymphocytes. Now, there are 3 subtypes of ILCs, appropriately named ILC 1, 2, and 3. ILC1 is a major producer of IFN gamma, which is a potent defense against viruses. ILC2 produces IL5, 9, and 13, which are used against helminths and allergic inflammation. And then ILC3 produces IL22 and interleukin, or IL17, which is used primarily in intestinal barrier functions. Now, these 3 cells all have transcription factors that help produce these cytokines. I'm not too interested in these because they don't pop up too often in the book. We do see T-bet a few other times in subsequent chapters, mainly when we talk about T lymphocytes. The other 2, not so much, so I'm less interested in those. But an important note about these cells is that they're directly analogous to 3 subtypes of helper T cells, otherwise called CD4 helper T cells. And these 3 different subtypes, TH1, 2, and 17, and I'm also, by the way, slightly annoyed that it doesn't quite follow the nice nomenclature of 1, 2, and 3. It has to go 1, 2, 17, and there's reasons for that, but annoying nonetheless. But what's really interesting about these T lymphocyte subclasses is how analogous they are to these innate lymphoid cells. Okay, they all exhibit the same transcription factor. They produce the same primary cytokines. So it's almost like a direct mirror of the innate immune system and adaptive immune system. Next, we're going to talk about natural killer cells. So we had previously described these as lymphocyte-like cells. They have similar lymphocyte morphology, and they're also derived from lymphocyte precursors. So they're really the first ILCs that we had described before. In fact, they're sometimes referred to as a type of ILC1 because of their secretion of IFN gamma. But they do constitute major differences between the other ILCs that we learned, like ILC1s, which are usually tissue located, yet natural killer cells constitute about 5 to 15% of all mononuclear cells in the blood and spleen. So they're very much unlike their ILC helper siblings. And then just a reminder, the fact that they have that natural designation helps put them in the innate immune response because they can act without differentiating any further, okay? Just like other macrophages or neutrophils, they're ready to go and already have program mechanisms built into them, and they don't need to have to adapt in order to carry out their killing functionalities. Now, natural killer cells can kill through two primary pathways. The first is through a cytokine-mediated pathway. So first, natural killer cells are stimulated by the interleukins 12, 15, and 18, and type 1 interferons, to stimulate the secretion expression of IFN gamma, right? So this will help induce macrophages to destroy their phagocytosed microbe. The second is through a cell-mediated pathway that's almost identical to the killer T cells, the CD8 plus cells of the adaptive immune response. Like CD8 plus cells, natural killer cells have granules. These are internal payloads of proteins that, when released and come into contact with infected cells, induce apoptotic or suicide mechanisms within that cell. There are two primary proteins here. The first is called perforin, and I like to think of it that perforin is meant to perforate the cell membranes of the target cells, which then allow the influx of granzymes, which is the second type of protein. And then granzymes are what initiate the sequence of apoptosis or suicide within the infected cells, directly killing them through the cell-mediated pathway. So one of the big takeaways here is that natural killer cells help act against microbes and pathogens that are in the intracellular compartment. Okay, so they're not actively killing bacteria or viruses in the extracellular space. It's cells that are already infected. Now, it's important to note that natural killer cells don't just go around killing cells. Okay, their killing action is regulated by a balance of activating receptors that sense infected and injured cells, and also inhibitory receptors, which bind to healthy normal cells. So two main classes of their activating receptors are killer cell immunoglobulin-like receptors, abbreviated KIRs, and then CD16. Now, CD16 binds weakly to the non-specific portion of an IgG antibody, okay, something we call the IFC portion, which is the stem of the Y. And we'll learn about that, I think, in Chapter 5. Now, because of this is a process known as antibody-dependent cell-mediated cytotoxicity. So when natural killer cells sense that particular IgG antibody, they know that something is wrong and there's an affected cell because otherwise that antibody wouldn't have been produced. And it initiates that killing action. On the other hand, inhibitory receptors on natural killer cells recognize a type of protein clusters on healthy cells called class 1 MHCs, which stand for major histocompatibility complexes. We're going to talk about MHCs a lot in future chapters because one of their primary roles, distinct from the regulations of natural killer cells, is their function displaying peptides and antigens from cytosolic proteins to CD8 plus T lymphocytes. So they're vitally important for activating CD8 plus lymphocytes. For now, it's just important to note that they're present in healthy cells, and also because they're genetically coded, they're very specific from one individual to the next. So even if you put an uninfected, healthy cell and transplant it into another person, natural killer cells would not be able to recognize that MHC and then would likely be activated to kill that healthy cell that's not genetically similar to the host cell. And it's because of this MHC self-recognition limitation that it's so difficult to find a match whenever someone's looking for an organ donor. So that kind of gives an indication of how unique these MHCs are from one person to the next. What's also important about this is that many viruses have, I think very cleverly, developed ways of hiding MHCs, right? MHCs, again, are the ways cells present antigens to the killer T cells, okay, the CD8 plus cells. So some viruses had found ways to instruct cells to down-regulate their expression of MHCs, thus avoiding cytotoxic T lymphocyte elimination, yet this makes them susceptible to natural killer cell elimination. So it's a nice balance between the natural killer cell of the innate immune system and the cytotoxic T killer cells of the adaptive immune system. Two other subtypes of the innate immune response that I like to talk about are classes of T and B lymphocytes and mast cells. Now, these T and B lymphocytes are unlike their adaptive cell siblings in that while they are structurally similar and they have T and B cell receptors, their antigen diversity is very limited. For the T cells, this includes the invariant natural killer T cells. So here I really want to make sure you don't confuse these with either natural killer cells or cytotoxic T killer cells, the CD8 plus cells. These are natural killer T cells. Okay, so they are of the T cell type, not the natural killer type, yet they are not full T cells of the killing variety. There's also the mucosa-associated invariant cells, and then the previously mentioned intraepithelial T cells. In Chapter 2, we also talked about two subclasses of B cells, B1 cells, which are fetus-derived, and marginal zone B cells, bone marrow-derived cells. Sorry, they reside in the bone marrow. Both these type of B cells produce antibodies. One's of, again, limited specificity, just like the T cells. And then these antibodies are also lower affinity. As we're learned, as B cells get further and further activated or more differentiated, they can be instructed to produce antibodies with higher and higher affinities that are more effective than these lower affinity subtypes. Now, we've already talked about mast cells before. We learned that they are almost the tissue version of basophils. So they're not circulating, they're found in tissues. In fact, they're found very close to blood vessels because one of their primary mechanisms of acting is to release vasodilators like histamine that dilate blood vessels and make capillaries more permeable. Okay, so while they're usually present in the skin, mucosal epithelium, and connective tissues, they're always present to blood vessels for that reason. All right, so that vasodilation results in rapid changes in the blood vessel structure, and it promotes acute inflammation when these granules are released. And furthermore, they can also secrete proteolytic enzymes that can kill bacteria and inactivate microbial toxins. Because of this, we think of mast cells, along with tissue-resident macrophages and dendritic cells that reside in the tissue, as sentinel cells, ones that are merely on the lookout and whose primary purpose isn't to simply eliminate the pathogen, but let the immune system know of a pathogen invasion. Next up, we're going to talk about the soluble molecules of the innate immune system. Now, these soluble molecules are very often called the humoral branch of innate immunity, just like how antibodies are called the humoral arm of adaptive immunity. Okay, and they sort of function in two major ways. First, they act as opsonins, which basically means molecules that bind and coat cells, pathogens, whatever, by binding to microbes, and it enhances the ability of macrophages and neutrophils in any phagocyte to phagocyte those microbes, right? This is actually what those cells like to attach to, as opposed to binding directly to the pathogens. Second, they promote inflammatory responses that not only activate phagocytes, but they help recruit and bring phagocytes to the site of infection so that they can also directly kill microbes. Now, the way I want us to think about the complement system, the complement, by the way, is the name of these soluble molecules, is proteins in the blood and tissues that are capable of acting like a runaway reaction. Okay, that is, they have an ability to react and then amplify themselves until they can build up sufficient concentrations to cause inflammation or opsonize these cells. Okay, but first they need to be activated because they're present in the blood at all times, and they don't normally react unless pathogens or something is wrong, something that necessitates an inflammatory reaction. So complement can be activated in three different ways. Okay, first we're going to talk about is the classical pathway. And FYI, it's only called the classical pathway because it was discovered first. The alternative pathway, which we're going to talk about second, is actually much older on the evolution scale, but it was discovered second. Anyways, the classical pathway actually works hand in hand with the adaptive immune response because the first in a series of lines of complement proteins called C1q has to first bind with the stem portion of antibodies. It's called the Fc portion that are already bound to cell surfaces. Once C1q is bound, then it can initiate the further activation down the line of the complement system. Now, there are proteins of the innate immune system like pentraxins, which we're going to learn about in the next slide, that can also bind to C1q and activate complement independent of the adaptive immune system, so independent of antibodies. The second pathway, called the alternative pathway, can actually use a complement protein C3 directly, not require and skip the C1q step, because C3 can bind to LPS or other bacterial or pathogen structures directly on the cell and kind of skip the initial steps, but it takes a recognition of foreign bodies in order to start this. So this is why C3 normally can't bind to healthy cells within a host body, because there's no cell surface that it can attach to and initiate that step. A third alternative pathway called the lectin pathway is very similar to the classical pathway because it uses a compound called mannose-binding lectin, or MBL, which acts identical to the C1q Fc structure in the classical pathway. Now, what's important to recognize in all three of these steps is that regardless of how they get activated, they all converge into building a complex protease called C3 convertase. And then from there, there's processes downstream of building C5, C6, all up until C9, which completes the complement system. Now again, all I want you to really take away from this point is that these are present in the blood. There's proteins that get destroyed, there's complexes that get made, and a lot of those products are involved in inflammation. And there's multiple ways of initiating that pathway. And again, think of the complement system as a runaway reaction of creating all of these steps, C1 to C9, while making inflammatory products throughout the entire process. Now, there's three other classes of soluble molecules that I'd like to briefly touch on. I say briefly because we don't hear about these too much more often in the rest of the book. So I've already mentioned petraxins because of their ability to participate in the classical pathway independent of antibodies. Three common petraxins are C-reactive protein, CRP, serum amyloid P, SAP, and then long pentraxin, PX3. So we see these compounds pop up in a number of different ways throughout the inflammatory response. First, plasma concentrations of a pentraxin like CRP are usually very low in healthy individuals, but during an inflammatory response can increase up to 1,000-fold in response to stimuli, such as through cytokines IL-6 and IL-1. So we can actually measure this and use this as an assay for measuring the inflammatory reaction. We can actually do the same thing with serum amyloid P, SAP, and then all three of these participate in binding with C1q in the classical pathway, again, independent of antibodies. Now, because of their involvement during the acute phase response of inflammation, or acute inflammation, we call these compounds acute phase reactants. Next up, we have colectins, which are proteins with a collagen-like tail and calcium-dependent C-lectin head. All right, now we've already mentioned one of these before, mannose-binding lectin, which is involved in the lectin pathway of complement. Two other popular ones are pulmonary surfactant proteins, SPA and SPD. Now, SPA and SPD are found in the alveoli of the lungs. They help reduce surface tension of mucus to allow for expansion. FYI, they reduce surface tension, therefore they are called surfactants, hence the SPA and SPD. They act as opsonins to facilitate ingestion by macrophages present there, and they also themselves directly inhibit microbial growth. Now, very similar to colectins are phacolins, but they substitute a C-lectin head with a fibrinogen-type carbohydrate domain. So to help me remember, I think F, fibrinogen, phacolin, and C, C-lectin, colectins. So to recap, the two big takeaways of these last two slides are the involvement of these molecules in opsonization and phagocytosis of phagocytes like macrophages and neutrophils, and also the role in the complement activation process, and then the complement system in general. So now we come to acute inflammation. I've said inflammation many times throughout this chapter, so let's first start by defining it. Okay, it's the accumulation of leukocytes, plasma proteins, and fluid derived from the blood at an extravascular tissue site of infection or injury. As we very much like to say, the first responders are typically neutrophils, in part because they're the most abundant leukocyte, and also they're the most rapid responder to chemotatic signals. Okay, the secondary responders are monocytes. Okay, these become macrophages in the tissue, and in fact, these become increasingly prominent over time. In fact, most inflammatory reactions tend to be dominated by monocytes/macrophages. Plasma proteins responsible in inflammation are complement proteins, antibodies, and acute phase reactants. There's also the reversible dilation of blood vessels, typically through a compound like histamine. This also helps make the vessels more permeable to water and fluid and allow fluid to leave and then form and collect at the extravascular tissue site. Duration can be as quickly as minutes to a few hours and last for a few days, after which the patterns start to change as it turns into chronic inflammation. Now, inflammation is initiated by the signals of pro-inflammatory cytokines, which are usually produced by tissue macrophages and dendritic cells to a large degree, although any other cells at the site of infection, like mast cells, endothelial and epithelial cells, are capable of secreting these as well. Now, their location, because they're exhibited or expressed by localized tissue tend to act on a paracrine action level, which means that they act on cells close to the origin of the inflammatory response. But in extreme cases, when inflammation is really severe, then these cytokines can act in an endocrine action, which means that sufficiently high concentrations can enter circulation and act in a systemic manner throughout the body. Now, as we're going to learn soon, many of these cytokines have similar or overlapping action. They can also be functionally unique in many ways, and they can also serve to amplify the expression of other cytokines in sort of a cascading and amplifying or feedback manner. Now, prominent cytokines of the innate immune cells like TNF, IL-17, IL-5, and IFN-gamma, in fact, I think we're going to be talking about all of these in subsequent slides, are also produced by T lymphocytes of the adaptive immune system. So it's not just acute inflammatory cytokines, they could also be adaptive immune cytokines as well. The first cytokine we're going to talk about in detail is tumor necrosis factor. Now, TNF originally got its name because it was originally identified as a serum substance that caused necrosis of tumors. And a quick word on the nomenclature, TNF is used interchangeably with TNF alpha. And that's really to differentiate it from a very similar cytokine that we're not going to talk about too much, TNF beta. So whenever someone says TNF, tumor necrosis factor, they're usually referring to the alpha variety of it. Now, cells have two distinct receptors for TNF alpha. Now, really, the only thing to highlight here is that both of the affinities to the receptors at TNF alpha is unusually low for a cytokine. Most other cytokines have affinities about an order of magnitude higher than this. TNF alpha is produced mainly by macrophages and dendritic cells. That covers around 90% of the TNF associated with the acute inflammatory response. But other cells, of course, are capable of secreting TNF alpha. The recognition of PAMPs and DAMPs via toll-like receptors, non-like receptors, and receptors in the cytosol all help induce TNF alpha gene expression. So common ones that would cause this are LPS via attachment to toll-like receptors, and then in gram-positive bacteria, lipoteichoic acid stimulate toll-like receptors. Now, tumor necrosis factor has a big role in a condition known as septic shock, which is the result of severe infections mediated in large part by TNF alpha. And because of this, there are many drugs for not just septic shock, but inflammatory diseases in general that are mediated through anti-tumor necrosis factor agents. The next cytokine to be discussed is IL-1, which stands for interleukin-1. As you may have noticed, many cytokines start with the IL format, and that's what that means, interleukin. So IL-1 is also a mediator of acute inflammatory response and has many similar actions as TNF alpha does. Now, there are two forms of IL-1 that are only 30% similar to each other, but yet still bind to the same receptors. There's IL-1 alpha and IL-1 beta. Now, like TNF, there's one that's commonly referred to, and TNF alpha are synonymous with each other. There's a very similar pattern to IL-1 in that whenever people usually say IL-1, they usually mean IL-1 beta. So in this case, it's not the alpha version, it's the beta version. So we just have to remember that it's sort of the opposite for IL-1 than it is for TNF alpha. Now, IL-1, just like TNF, is produced mainly by macrophages and dendritic cells, but unlike TNF alpha, many other cells not just are capable of producing IL-1, but commonly produce it as well. Now, it's interesting to note that TNF alpha itself can stimulate the production of IL-1. And this is sort of an example of the cascading systems of cytokines, how one can feed in to the other, even though they're both performing very similar actions. This helps amplify the inflammatory response. Now, there are two receptor types for IL-1. The common one, type 1 IL-1 receptor, is expressed on many different cell types. The type 2 IL-1 receptor is incapable of activating downstream signaling for the inflammatory response, and it's thought to act as a decoy to limit IL-1 responses. So in cases where inflammation is no longer necessary, this receptor might be overexpressed, whereas the type 1 version might be underexpressed. So this helps to act as a feedback system to limit inflammation and keep it in check. Next, we're going to talk about IL-6. Now, I wonder if you've caught the pattern so far, but if not, I'm going to repeat it again. IL-6 is another mediator of acute inflammatory response. These ones have both acute and systemic effects. So IL-6 functions to induce synthesis of the acute phase reactants in the liver. It stimulates neutrophil production and also promotes differentiation of a subtype of CD4+. These are the helper T lymphocyte cells Th17 in particular, which also functions to work directly with neutrophils, as we're going to learn about later on down the road, so you don't need to know that right now. They're produced mainly by mononuclear phagocytes, and whenever I say that, I want you to think monocytes, macrophages, and dendritic cells. Endothelial cells, fibroblasts, and then other cells in response to PAMPs and IL-1 and TNF. So this is another example of that cascading effects where the expression of one cytokine feeds into the expression of another cytokine. IL-6 is also a major contributor to inflammation in several human inflammatory diseases, notably rheumatoid arthritis. And because of this, antibodies specific for either IL-6 or the IL-6 receptor are used to treat arthritis. Now, as you can imagine, there are many other different types of cytokines, and I'm just going to list them here for the sake of brevity. And I think it might be better that we learn about them as we talk about their functions within that context. Some of the ones I have listed here are IL-12, which is used in the differentiation of the Th1 subtype of helper T cells. It also has roles in inflammatory bowel disease and psoriasis. IL-T helps enhance the function of natural killer cells. IL-25, another cascading cytokines that promote cells to produce IL-4, IL-5, and IL-13. The list goes on. So now let's learn how these cytokines are used in leukocyte recruitment. So as we learn, in order for leukocytes to be recruited to the site of inflammation, endothelial cells have to start expressing E-selectins and then increase their expression of integrins for leukocytes. And TNF and IL-1 both function to do just that. They help these cells express E-selectin and increase expression of those integrins. Furthermore, these two cytokines also stimulate cells to secrete chemokines that bind to cell receptors. So for instance, we learned that neutrophils are the first responders due to an abundance, particularly at a very early stage, of the chemokine CXCL8. TNF and IL-1 are responsible for that. And it's the same for monocytes and their respective ligands CCL2. But they're not just involved in recruitment, because TNF and IL-1, along with IL-6, enter circulation and enhance the production of neutrophils in bone marrow as well. So next up, we're going to talk about phagocytosis. Now, we've mentioned this I think in every chapter up to this at this point, but I'm going to define again. Phagocytosis is the process of cell eating, where cells ingest other cells by a process of pinching off the lipid membrane to form intracellular vesicles. We've already talked about a number of cells that classify as phagocytes. Classically, neutrophils and macrophages are the ones I want you to think of when we talk about phagocytes. Of course, other cells are capable of phagocytosing, like dendritic cells, mast cells, and basophils. Now, the first step in phagocytosis is just binding via specific receptors on the phagocyte cell membrane. These could be through pattern recognition receptors, but this first requires that those microorganisms express those particular patterns specific to those receptors. There's also receptors for opsonins, including antibodies, complement proteins, and plasma lectins. Now, the heavy-hitting receptor, the one that's most potent for phagocytosis, is Fc gamma R1, which means that is specific to an Fc portion of an antibody. And again, I mentioned a couple of times, Fc is the stem portion of an antibody. I say this is potent and a heavy-hitting receptor because antibodies are the, quote, most efficient systems for opsonization, and thus phagocytosis. So if an individual responds to an infection by making IgG antibodies against microbial antigens, the IgG molecules bind to these antigens. The Fc ends of the bound antibodies can interact with the Fc gamma R1 receptor on phagocytes, and the end result is a very efficient and potent form of phagocytosis of the microbes. So this is just another example of the link between innate and adaptive immunity. So far on the slide, we've really only covered that very top left-hand portion of that drawing, but the end result of phagocytosis is a phagosome. Right? This is just a vesicle formed around pieces of the plasma membrane that engulfs a foreign particle, ingests it, and breaking away into the intracellular compartment. Once there, phagosomes fuse with lysosomes to create phagolysosomes, and this is where most of the killing action occurs. Now, there's three primary chemicals that are used in this killing mechanism. There are reactive oxygen species, primarily used by neutrophils, other macrophages also to a lesser degree. Now, these ROS are highly reactive oxidizing agents. They're stimulated by IFN gamma and signals from toll-like receptors. and it requires oxygens through cellular respiration. Okay, this is a mechanism known as respiratory burst. There's also nitric oxide, which is more on the macrophage cell type. This is also stimulated by FN gamma signals from toll-like receptors. Furthermore, there's a cooperative function between these two chemicals, in that nitric oxide can combine with hydrogen peroxide or superoxide, both reactive oxygen species, to produce a highly reactive peroxynitrite radicals that can also help kill microbes. Furthermore, there exists proteolytic enzymes such as elastase or cathepsin that can help destroy microbes as well within these phagolysosomes. Now, one really cool side note that I want to talk about is the ability in neutrophils to extrude their DNA and granule contents to form neutrophil extracellular traps. This is in the extracellular space that can trap and kill bacteria. Now, of course, this also kills neutrophils, but it's a pretty gnarly way of committing suicide for the sake of killing microbes. Now, while inflammation certainly has important roles in the defense of our body, there's also systemic and pathological consequences to inflammation. So for instance, TNF, that's TNF alpha again, and IL-1 both act on the hypothalamic in the brain to produce fever. So because of this, they're known as endogenous pyrogens. Pyrogen means to induce fever. This is to contrast them with exogenous pyrogens like LPS. Now, an anti-fever agent like aspirin reduces fever by blocking the hypothalamic enzymes stimulated by TNF and IL-1. So while they don't act on these cytokines directly, they do so indirectly by the enzymes that are stimulated by these cytokines. Hepatocytes in the liver are also stimulated by IL-1 and IL-6 to produce acute phase reactants. We already talked about these, CRPs, SAPs, fibrinogen. And the big one, which I've actually also mentioned previously, is TNF and its role in septic shock. So TNF-alpha inhibits myocardial contractility, which decreases blood pressure. It causes intravascular thrombosis by impairing normal anticoagulants, so that's the formation of blood clots. Prolonged TNF exposure causes wasting of muscles and fat cells. And then the big one is sepsis, right? So this can be initiated by LPS, a lipotecaleic acid, to eventually produce septic shock, where your body eventually goes into shutdown. It's one of the main reasons for death in hospitals. Next up, we're going to talk about the innate immune system's antiviral response. So the primary mechanism for fighting against viruses is the production of type 1 interferons, also abbreviated IFN. These compounds are named so because they interfere with viral replication. Two primary subclasses of IFNs, of which there are many subclasses, by the way, but the two ones we're going to be concentrating on are IFN alpha, of which there are 13 different types, and IFN beta, which there's only a single type. It's IFN beta. Plasmacytoid dendritic cells are a major source of IFN alphas. The most potent stimuli for IFN are viral nucleic acids. So if you remember back when we talked about the pattern recognition receptors, now we're thinking of the RIG-like receptors and the intracellular toll-like receptors. If I remember correctly, those are numbers 3, 7, 8, and 9. I also have to remember that the 3 is the odd one out, and then 7, 8, 9. Now, type 1 interferons act in a number of different ways to help protect against viral infections. The first is that they stimulate production of IFN to activate transcription factors in neighboring cells. So this is a paracrine action as opposed to acting upon themselves. So they're sort of sacrificing themselves in order to instruct their neighbors on how to act. Now, these neighboring cells are told to enter what's called an antiviral state, in which they block RNA transcriptions, which is the replication factors that viruses typically take use of, and they also promote RNA degradation. Furthermore, type 1 interferons help promote the migration of lymphocytes into lymph nodes by inducing the expression of CD69. Now, if you remember from the previous chapter, CD69 binds with a complex of surface receptors S1P-R1, in a way suppressing it so that these lymphocytes stay within the lymph nodes longer. Type 1 interferons also help to promote the effectiveness and cytotoxicity of both natural killer cells and the CD8 plus cytotoxic T killer cells. They promote the differentiation of the type Th1 subtype of CD4 plus cells. Those are helper T cells that are involved in phagocytosis. Type 1 IFNs also upregulate class 1 MHCs. And I mentioned this in, I think, this lecture or previous lecture, but MHCs are major histopathological complexes, which are vital for presenting antigens to CD8 plus cells. Again, these are the exact killing cells that you would need, particularly for viral replication, so cells that have already been effective and have intracellular pathogens. And because of these benefits, interferon alpha is used to treat some viruses like viral hepatitis, and also some tumors because of its promotion of CD8 plus cells, which are very useful in fighting tumors and cancer. And IFN beta is used to treat and limit attacks in multiple sclerosis as well, although the mechanisms here are not as well understood. Now, to close out this chapter, I just want to talk about the innate system's ability to stimulate the response of adaptive immunity. Now, the activation of lymphocytes isn't just simply a recognition of an antigen. It requires two distinct signals. One is that response of an antigen, like through a T cell or a B cell receptor. And the second signal is a response from innate immunity stimuli. This is called the two-signal hypothesis. Now, the first signal ensures that an immune response is specific to that particular antigen. The second signal ensures that the adaptive immune response is induced by a dangerous infection and not just harmless antigens, for instance, like through self-antigens when there is no infection. Furthermore, the second signal doesn't just enhance the magnitude of the adaptive immune response, but can also influence the nature of it as well. For instance, dendritic cells and macrophages can encounter microbes in the intracellular space, secrete cytokines, and present those antigens to T cells, and mediate macrophages to kill and phagocytose those microbes. For microbes that are in the extracellular space, like in the blood, they could encounter the complement pathway, which could enhance the production of B cell antibodies. These then opsonize the microbes and again promote phagocytosis. Now, it's because of this that whenever vaccines are developed, they don't just have the antigen, the purified protein antigens in the mixture. They require what are known as adjuvants, something to create that signal 2 in addition to that signal 1 to promote lymphocyte activation. Now, in closing out our chapter on inflammation, I'd like to talk about the regulatory mechanisms that are meant to inhibit inflammation, right? Because it's important to note that the innate immune response is self-regulating. So one cytokine that's involved in this is IL-10, which is produced by and inhibits activation of macrophage and dendritic cells. IL-10 serves to reduce the secretion of all the inflammatory cytokines we just talked about, so IL-1, TNF-alpha, IL-12, and because it's both produced by and affects macrophages and dendritic cells, it's an excellent example of a negative feedback regulator. There also exists an antagonist to IL-1 receptor, appropriately named IL-1 receptor antagonist, or IL-1RA, that is structurally homologous to IL-1, but is biologically inactive. So by competing with it and not doing anything, it blocks that receptor from being activated by IL-1 itself. And because of that, recombinant versions of this antagonist is used for treatments of familial fever syndromes and rheumatoid arthritis. And lastly, I'll mention the type 2 IL-1 receptor, which I mentioned previously when we were talking about IL-1, that acts as a decoy receptor because it can compete for IL-1, it can bind for it, yet it doesn't initiate any downstream signaling processes that IL-1 does. So you can imagine situations where either the body might overexpress this decoy receptor, or even drugs might initiate the overexpression of this receptor in order to downregulate the role of IL-1. All right, that's it. Hope you enjoyed it.